Warm greetings from Rock City, Tiruchirappalli and Harshamitra Super Specialty Cancer Center, Trichy. We are going to discuss about the molecular approaches in breast cancer. Individualized treatment is the order of the day in breast cancer management. Treatment has to be tailored according to molecular subtypes based on gene array studies or approximated to ERPR, HER2 and Ki67 based on immunohistochemistry. Before we get into the depths of molecular genetics in breast cancer, we'll quickly go through the basics of cellular biology. The cells in our body are under tight regulation. They grow, divide, and after some divisions, stop dividing and undergo cell death, a process called as apoptosis. But cancer cells do not follow these rules. They divide continuously, they lack contact inhibition, and they lack apoptosis. This orderly process of cell cycle is regulated by the nucleus, which is the brain of the cell, that contains all the information packed in chromosomes. There are 23 pairs of chromosomes in each cell. Chromosomes are nothing but tightly coiled structures of long chain of DNA wound around a protein called histone. A segment of DNA that codes for a specific protein is called as gene. Gene is nothing but a short segment of DNA that can produce a protein. Various cellular events are controlled by these proteins produced by the gene when appropriate signal is received. Cells receive signals and they communicate with each other through signals. Signals are received by cells through receptors on their surface. The signal molecules, called ligands, bind to the receptors and activate the receptors by a process called dimerization and phosphorylation. This activates second messengers within the cell. A cascade of downstream events take place and intermediate molecules are produced. The intermediate molecule enters the nucleus and activates the transcription factor. Transcription factor reaches the DNA. It scans the entire DNA and gets attached to a specific gene site. The transcription factor then gets activated. Co-regulatory proteins join and the process of transcription begins. DNA gets cleaved to form mRNA, which codes for a protein. Non-coding portion of mRNA, called as microRNA, is lost in the process and only the coding portion leaves the nucleus and enters the cytoplasm. It gets attached to ribosomes and with the help of transfer RNA forms a protein. Protein, which is a long chain of amino acids. The protein thus formed brings about the various cellular events. A protein formed as a result of activation of the receptor and activation of downstream pathways controls the cell. The basics of cellular genetics is very important in cancer genetics because therapeutic interventions target these areas. It is important to understand this sequence of events from the receptors to activation of pathways to activation of gene and production of the protein. The function of the protein thus produced depends on the nature of the gene. In cancer genetics, we are interested in two balanced groups of genes, tumor suppressor genes which calm the cell and proto-oncogenes that activate the cell. When a signal is received, the proto-oncogene gets activated and cells grow and divide as in normal growth of an individual. Any abnormal growth will be checked by the tumor suppressor gene. But mutation in one of these genes will topple this balance and the cell gains a growth advantage. which results in proliferation of the cells. Abnormal proliferation of ductal epithelial cells occur due to mutations that can be acquired or inherited. 90% of the mutations are acquired and 10% of them are inherited germline mutations. BRCA1 and BRCA2 are the commonest. BRCA1 and BRCA2 genes are present on chromosomes 17 and 13. More than 100 mutations as single nucleotide polymorphisms have been identified, and these SNPs confer a lifetime risk of 85% in developing breast cancer in BRCA mutated women. These BRCA mutated women, when they develop breast cancer, they are young and the tumors are aggressive. The tumors are triple negative or basal molecular subtype. They are ERPR negative and HER2 negative. They are aggressive and difficult to treat. Understanding the molecular genetics has now shown that certain drugs are more effective in this triple negative molecular subtype. BRCA genes are basically DNA repair genes. Tumors arising in BRCA mutation cannot repair their DNA. Hence, drugs that cause DNA damage are more effective in these tumors. Cisplatin, for example, which causes double-strand breaks, is found to be more effective on these triple-negative tumor cells because the tumor cells cannot repair the DNA damage caused by cisplatin. The tumor tries to use alternate PARP-mediated single-strand repair. PARP inhibitors like Clinparza prevent this repair and are effective in not only BRCA-related triple-negative breast cancers but also acquired triple-negative breast cancers. 90% of the breast cancers are due to acquired mutations. About 50 to 80 mutations are seen in each tumor. These mutations are studied using microarray technology. When a number of tumors are studied, some genes are common to almost all tumors, forming gene mountains in the genomic landscape. But these are few in number, like p53 mutation or AKT mutation. There are many uncommon genes forming gene hills which are different for different tumors. This genomic landscape of a tumor is called as molecular signature. Tumors with similar molecular signature are grouped together. Thus, we get 5 different molecular subtypes: luminal A, luminal B, HER2 enriched, triple negative or basal type, and normal like type. These types differ in their gene expression profile and hence their clinical behavior. For example, p53 mutation is high in aggressive types like HER2 enriched or triple negative types, but it is low in luminal types. Luminal type is the most common. Luminal A is ERPR positive and HER2 negative and has the best prognosis. A subset of node negative patients in this luminal group can even avoid chemotherapy. Molecular genetics helps in treatment decisions. 21 gene analysis called Oncotype DX helps to identify the low risk category that can avoid chemotherapy and be managed with hormone therapy alone. Oncotype DX can also be combined with pathological features to give a better prognostic integrated score. Avaria DX is another prognostic or predictive factor. These luminal tumors express estrogen receptors. When estradiol binds to these receptors, the AF1 and AF2 sites of the receptors get activated and dimers are formed. These dimers activate transcription and produce growth proteins, causing tumor proliferation. As we have already seen, these dimers go and get attached to the estrogen responsive site in the DNA, co-regulatory proteins join, and the process of transcription begins to produce growth factors that stimulate the growth of the tumor cells. Ovarian ablation and aromatase inhibitors act by reducing the estradiol level and thus reducing the growth proteins. On the other hand, selective estrogen receptor modulators like tamoxifen, which are depicted by the blue colored molecules, bind to the receptors and modify them so that only AF1 site is activated. AF2 is not activated. This partial agonistic activity results in less amount of growth proteins. The transcription is only partly active. The dimers are only partly activated. This results in reduced growth proteins. Fulvestrant is a drug that binds to the receptors but does not activate them at all. Both the AF1 and AF2 sites are not activated. Very few dimers are formed. Receptors which do not dimerize get destroyed. Fulvestrant causes total blockage of transcription. The other molecular subtype is HER2-enriched, seen in 20% of breast cancers. It has poor prognosis but responds to target therapy. HER2 receptors belong to the EGFR family. HER2 is unique that it does not bind to ligands. They get activated by joining with HER1 or HER3 to activate many pathways that make tumor cells grow. These receptors are overexpressed in HER2 positive subtype and signal for cell proliferation is enormously high. Target agents like trastuzumab bind to these receptors and block the downstream signals, causing tumor cell death by various mechanisms, out of which binding to the receptors is the main mechanism. Pertuzumab is another monoclonal antibody that binds to domain 2 of HER2 and can be used along with trastuzumab, which binds to domain 1 and have a synergistic effect. When the ligand binds to HER3 receptors, for example, dimerization of HER2 with HER3 is blocked by these monoclonal antibodies and hence downstream pathway is not activated. Trastuzumab can also be combined with chemotherapeutic agent like maytansine to combine the targeting effect and cytotoxic effect. When the antibody drug conjugate reaches the cell, it gets internalized and the tumor cells get destroyed due to the cytotoxic effect. Interestingly, HER2 receptors can directly activate estrogen receptors. This crosstalk between the receptors explains tamoxifen resistance in HER2 positive tumors. Aromatase inhibitors or fulvestrant to be used in these HER2 positive group. Molecular genetics not only helps in treatment decisions but also prognostication. Gene signatures like MAMA print and Rotterdam signatures are used for prognosis. Like bacterial culture to find antibiotic sensitivity, do we have tumor cultures to find chemosensitivity? Unfortunately, no. Molecular genetics may have an answer for this. Multigene analysis of FNAC specimen to predict response to taxane FAC regime is under study. From cancer genetics, we now move on to cancer epigenetics. We know that gene mutations result in tumor formation. Epigenetic literally means on top of the DNA. A methyl group on top of the DNA can indirectly control the gene. Increased methylation of DNA can activate the gene and can cause mutations and result in tumor formation. We saw that DNA is coiled around a protein called histone. When acetyl groups attach to histone, unwinding of DNA occurs, resulting in mutations. Hence, drugs that inhibit DNA methylation and histone acetylation are being studied. These drugs, DNA methyltransferase inhibitors and histone deacetylase inhibitors, are under study. Epigenetics plays a role in cancer detection also. Detection of methylated DNA sequences in peripheral blood can help in early detection. A very hot area of research in epigenetics is microRNAs, small non-coding RNAs. During the process of transcription, where DNA is converted to mRNA, part of the non-coding portion is lost, and this is called microRNA. MicroRNAs were considered to be non-functional, but now we know that they can bind to complementary mRNAs and prevent protein formation. All these genetic and epigenetic events use pathways to stimulate the cell. Pathway inhibition is another novel concept in molecular genetics. Notable among them are the estrogen receptor pathway, which we have already seen, and the others are RAS pathway and phosphoinositol pathway. There are targeting agents that can interfere with this pathway at various levels. Drugs like farnesyl transferase inhibitor, which can inhibit RAS, or drugs like mTOR inhibitors, temsirolimus, act by interfering with these pathways. Angiogenetic inhibitors like bevacizumab, which is a vascular endothelial growth factor inhibitor, have no proven role in breast cancer. To summarize, the clinical application of molecular genetics is in prevention, treatment, prognosis, and personalized care. The impact of a gene test on clinical practice is best emphasized by the double mastectomy of Angelina Jolie due to BRCA mutation. Gene tests like Oncotype DX can be used to avoid chemotherapy in selected subsets. Traditional folk prognostic factors are necessary but not sufficient. We need gene array to tailor the treatment according to the molecular subtypes and personalize cancer treatment. To conclude, the treatment outcome of cancer patients depends on three factors: the tumor, host, and treatment factors. molecular genetics plays a central role, and we still have a long way to go. I thank my technical persons. I thank my colleagues in the biotechnology department in Bharatdasan University. And thank you all viewers for seeing this. Thank you.